How do our memories last for a lifetime when the very molecules that encode them are replaced every few days or weeks? This age-old puzzle, first articulated by Francis Crick in 1984, has driven neuroscientists to search for the elusive molecular basis of memory persistence. The story of Todd Sacktor, a neuroscientist whose own childhood memory of his late sister lingers after 6 decades, serves as both a personal and scientific entry point into this mystery. Sacktor's quest began early in his career, inspired by the notion that every memory is a physical trace etched into the brain's neural fabric. At SUNY Downstate in Brooklyn, he set out to identify the molecular mechanisms that allow memories to persist despite the constant turnover of cellular components. The prevailing theory, rooted in Donald Hebb's 1949 insight, cells that fire together, wire together, suggested that the strengthening of synaptic connections between neurons underlies memory formation and retention. In the early 1990s, Sacktor's experiments on rat hippocampal slices led to the discovery of a protein called protein kinase Zeta, PKMζ. He observed that stimulating neural pathways to mimic memory formation consistently increased PKMζ levels in synapses. Subsequent experiments showed that blocking PKMζ activity could reverse synaptic strengthening and even erase established memories, suggesting that PKMζ was both necessary and sufficient for the maintenance of long-term memory. The scientific community initially embraced PKMζ as a molecular key to memory. Studies worldwide confirmed that inhibiting PKMζ could erase various types of memories in animals. However, the narrative faltered when genetic studies in 2013 revealed that mice lacking PKMζ could still form memories, casting doubt on its singular role. Sacktor and his collaborator Andre Fenton responded by demonstrating that in the absence of PKMζ, other molecules compensate to preserve synaptic strength, highlighting the resilience and redundancy of biological systems. The turning point came with the discovery of KIBRA, a scaffolding protein abundant in brain regions involved in learning and memory. Researchers found that KIBRA binds to PKMζ, anchoring it at synapses. This persistent bond maintains synaptic strength even as the individual proteins are degraded, and replaced a molecular relay that preserves the memory trace. Disrupting the KIBRA-PKMζ complex in mice not only erased established memories but also prevented their spontaneous recovery, underscoring the complex's critical role in both the formation and maintenance of long-term memories. This discovery elegantly addresses Crick's paradox, which postulates memories can outlast the molecules that encode them because the KIBRA-PKMζ complex acts as a molecular glue, perpetuating the memory trace through continued renewal. KIBRA also appears to serve as a synaptic tag, guiding PKMζ to the precise synapses involved in specific memories, ensuring that only relevant connections are strengthened. While the KIBRA-PKMζ partnership represents a major advance, some scientists, like David Glanzman, caution that synaptic strengthening may not be the sole mechanism of memory storage. Alternative models propose that molecular changes within neurons themselves may also encode memories. Nevertheless, the discovery of the KIBRA-PKMζ complex provides a compelling answer to how memories persist in a dynamic, ever-changing brain, and opens new avenues for understanding and potentially treating memory-related disorders. This molecular dance of memory, where fleeting proteins form enduring partnerships, reveals the astonishing complexity and resilience of the human mind.